The topic selected for today is respiration in plants or cellular respiration. Though we call it as respiration in plants, the same mechanism occurs in all the living animals also. Then, coming to the definition of respiration, respiration is the process by which chemical energy stored in the organic molecules is released by oxidation. Then oxidation occurs in three ways. It occurs by addition of oxygen or removal of hydrogen, and also by the removal of electrons. For example, there are hydrogen carriers like NAD and FAD. In the presence of the enzyme dehydrogenase, they combine with hydrogen ions. Similarly, if electrons are removed from a molecule or from an atom, it is also known as oxidation. In this way, oxidation occurs in three ways. Then, coming to the process of respiration, it is two types: aerobic respiration and anaerobic respiration. Aerobic respiration does not require any oxygen, whereas aerobic respiration requires oxygen. Anaerobic respiration is a very primitive one, and this is present in all the living beings. When the atmosphere appeared on Earth, it was a reducing one. There was no oxygen in the atmosphere. So the primitive organisms, when evolved, they could not use any oxygen. So they have adopted anaerobic respiration. That means respiration without oxygen. This anaerobic respiration is known as glycolysis. And glycolysis occurs in the cytoplasm. Later on, oxygen got accumulated in the atmosphere due to the action of the green plants and cyanobacteria, and aerobic respiration also evolved. Even after the appearance of aerobic respiration, anaerobic respiration is still maintained. Even today, anaerobic respiration occurs in all the living beings. There is no exception, and it always takes place in the cytoplasm. Then, from where we get the energy to run the metabolic processes? See, to run any metabolic process, energy is required. So, the energy that runs all the metabolic processes in all the living beings comes only from the sun, except in the case of some chemoautotrophs like sulfur bacteria or iron bacteria. So, almost the universal source of energy on Earth is sun. Sun releases vast amounts of energy in the form of radiant energy. This radiant energy is trapped by the green plants in their chlorophyll, and this energy coming from the sun is stored in the form of bond energy. First in the ATP, I have explained all these processes in the photosynthesis. Then from ATP, various types of carbohydrates, proteins and fats are synthesized. In this way, the energy coming from the sun is trapped by the green plants and stored in the form of energy. So whenever we require energy, or whenever the living organisms require energy, this energy stored in the organic compounds is released. If all the energy is released at once, in one step, what happens? Much of the energy is lost in the form of heat. So to avoid this loss, the respiratory process is a slow and stepwise fashion. As a result, energy is released in small amounts in each step or in different steps. And this energy is picked up by ADP and it is converted into ATP. That means the energy is stored in the form of ATP, and this energy is used for various metabolic activities. So ATP is known as energy currency, and it is rechargeable. Whenever ATP becomes ADP, ADP is again becomes ATP. So, like a battery, ATP is a rechargeable substance. In ATP, there are three phosphate molecules. ATP means adenosine triphosphate. The last two phosphate molecules, as seen here, in adenosine triphosphate, are high energy bonds, these two. When this bond is broken, the phosphate is released and ATP becomes ADP. When this bond is broken, ADP becomes AMP, adenosine monophosphate, and some more energy is released. In this way, ATP has two high energy bonds. Then, coming to again living beings, some are anaerobic, and these are either obligate anaerobic organisms or facultative anaerobic organisms. Obligate anaerobic organisms, they always depend on glycolysis only. Only anaerobic respiration occurs in them. In the case of facultative anaerobic organisms, what happens is, whenever oxygen is available, they carry out Krebs cycle, and when oxygen is not available, they carry out glycolysis. Glycolysis is a Greek term. Glycos means sugar, lysis means breakdown. So in the process of glycolysis, sugar is broken down. And I will explain that point later on. Then, the organic compounds are used in cellular respiration, and the substances generally used are carbohydrates. And the starting point in glycolysis is always glucose. If glucose and glycogen are exhausted, then fatty acids are employed. When fats are broken down into fatty acids and glycerol, the fatty acids provide energy and they enter the glycolytic cycle. The proteins are used only as a last option because proteins serve a variety of functions in the body. That's why proteins are rarely used in cellular respiration. Then, if the energy for phosphorylation means synthesis of ATP from ADP by addition of one inorganic phosphate ion. So, whenever ATP is synthesized from ADP by using energy and inorganic phosphate, it is known as phosphorylation. This phosphorylation is two types. If energy for phosphorylation comes from the sun, it is known as photophosphorylation. Photo means light. If the energy for phosphorylation comes from oxygen, it is known as oxidative phosphorylation. Then, removal of carbon dioxide is known as decarboxylation. So, in the process of respiration, oxidative phosphorylation occurs and also decarboxylation occurs. Then formula for glucose is C6H12O6. In the process of respiration, the carbon present in the glucose is lost in the form of carbon dioxide and it is expelled or used by the plants in photosynthesis. Oxygen combines with hydrogen and it is removed as water. Whereas the hydrogen ions are transferred to hydrogen carriers, NAD or FAD. These hydrogen carriers enter electron transport chain present in the cristae of mitochondria, where much of the energy is derived from the food substances. So in this way, the hydrogen carriers NAD and FAD are very important in the generation of ATP or in oxidative phosphorylation. Now let us take up the process of glycolysis. Remember, in respiration, glycolysis is the first step. In aerobic forms, there is another respiration known as Krebs cycle. or TCA cycle or citric acid cycle. Now, let us confine ourselves to only glycolysis, which occurs in all the living beings. And glycolysis takes place only in the cytoplasm, and it occurs in 10 steps. And each step is controlled by or catalyzed by one or more enzymes. I am not giving any enzymes here because it is not given in your textbook. Now let us see how the glycolysis proceeds in the cytoplasm. Glucose is the starting point. Glucose is a 6-carbon molecule. And remember one more point. There are 3 important steps in glycolysis. Glycolysis. First one is phosphorylation. Phosphorylation means the activation of sugar to make it more reactive. So when the phosphate groups are added, the sugar becomes more active and participates in reactions. Lysis. This is splitting or breaking off the 6 sugar into 2 3C sugars. The third step is oxidative dehydrogenation. That means here oxidation is carried out by the removal of hydrogen ions. These hydrogen ions are transferred to hydrogen carriers, NAD. So these are the 3 important steps. Then ATP is also synthesized. I will come to that point later on. The first step is glucose. It is a 6-carbon molecule and it is converted into glucose 6-phosphate by using one ATP molecule. So here ATP is converted into ADP. The phosphate goes to the glucose. So it becomes glucose 6-phosphate. Then by using another ATP molecule, the glucose 6-phosphate becomes fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Here there are 2 phosphate atoms here. Then this is called phosphorylation. The next step is lysis. Now the fructose 1,6-bisphosphate cleaves into 2 molecules of 3 carbons. One is glyceraldehyde 3-phosphate or 3-phosphoglyceraldehyde. The second one is dihydroxyacetone phosphate. These 2 are 3-carbon compounds or molecules. Then these 2 are interconvertible. This can become this one or this become this one. Then the dihydroxyacetone phosphate is also converted into glyceraldehyde 3-phosphate, and the glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglyceric acid or BPGA. Here onwards we have to take into account 2 molecules because the 6-carbon compound is broken down to 3 carbon compounds of 2. So we have to multiply by 2 all these steps. So the 3-phosphoglyceraldehyde is converted into 1,3-bisphosphoglyceric acid or BPGA. When this 1,3-bisphosphoglyceric acid is converted into 3-phosphoglyceric acid, one ATP molecule is synthesized. So this is the gain, one ATP molecule here. That means there are 2 molecules. So total 2 ATP molecules we get here. When the 3-phosphoglyceric acid is converted into 2-phosphoglyceric acid, then 2-phosphoglyceric acid is converted into phosphoenol pyruvic acid. During this step, 1 molecule of water is lost. Then phosphoenol pyruvic acid, which is also known as PEP, is converted into pyruvic acid. In this process, 1 more ATP molecule is generated. That means totally 2 more ATP molecules are generated. That means 2 ATP molecules are generated here and 2 more ATP molecules are generated here. Altogether, 4 ATP molecules are generated. Then, in the beginning, there is expenditure of 2 ATP molecules. So the net gain is 4 minus 2 ATP molecules. So this is the total gain of glycolysis. At the end of glycolysis, the net gain is 2 ATP molecules. Then the pyruvic acid is the end product of glycolysis. This pyruvic acid, either it becomes the Krebs cycle in the aerobic respiration, or it forms the ethanol by anaerobic respiration in the yeast, or it forms pyruvic acid in the muscles due to anaerobic respiration. So the fate of pyruvic acid is in three ways. It enters the aerobic respiration or the Krebs cycle and forms more and more energy, or it forms ethanol in anaerobic forms like yeast, or it forms the pyruvic acid in the muscles of animals. Now let us take up, it has entered the Krebs cycle. So pyruvic acid, what happens is, it enters the mitochondrial matrix. It is formed in the cytoplasm. But for further activities, it enters the mitochondrial matrix, and it is converted into acetyl group. So in the case of oxidative respiration, what happens is, the pyruvic acid enters the mitochondrial matrix and produces acetyl group, CH3COO. This acetyl group is picked up by coenzyme A. This coenzyme, after becoming the acetyl group, becomes acetyl coenzyme A, or simply acetyl CoA. This one. So in the mitochondrial matrix, pyruvic acid is converted into acetyl CoA. During this process, decarboxylation takes place. That means the 3 carbon atom pyruvic acid becomes 2 carbon atom acetyl CoA. That means 1 carbon atom is lost, and that is converted into carbon dioxide. This is called decarboxylation. Then 2 hydrogen ions are also removed, and they are accepted by hydrogen carriers NAD. In this way, dehydrogenation also takes place. So when pyruvic acid is converted to acetyl CoA in the mitochondrial matrix, decarboxylation and dehydrogenation takes place. Now, coming to the formation of NADH. In this step also NADH is formed. So before the glucose enters the Krebs cycle, how many NADH are formed? 2 here, 2 here, 4. 4 NADH molecules are formed. These NADH molecules enter the electron transport chain present in the cristae of mitochondria. There, each NADH molecule produces 3 ATP molecules. How it happens, I will explain when you take up the electron transport chain. So in this way, at the end of glycolysis, we get pyruvic acid, and the pyruvic acid has 3 types of fates. Either it becomes ethanol in yeast, or it becomes pyruvic acid, or it becomes the acetyl CoA to enter the Krebs cycle. So that is about first part of respiration plants, glycolysis. In the next class, I will take up the Krebs cycle. Thank you.